Well, hello everyone. Welcome to episode 2 of HEADCHECK Health Talks. We're excited to introduce your guest today, Sophie Prosser from the University of Birmingham. And so today we're going to just get Sophie to introduce herself, tell us a little bit about her background and her latest research. Then we're going to dive into that research a little bit, ask a few questions about that. And then to finish things off, we're just going to ask her a few questions about her clinic in general and how HEADCHECK has been, a little bit about how HEADCHECK has been working with them in the clinic. So to get started off, Sophie, thanks so much for joining us. Tell us a little bit about yourself, your background, and your latest research on biomarker variability in university athletes following concussion.
Yeah, so I'm a research fellow at the University of Birmingham. My research focuses mainly around areas of brain health. An area in particular that I focus on is biomarkers, another area being the vestibular system as well. I do a lot of work with concussion, do a lot of work with people with balance conditions. So I'm working on a clinical trial as well at the moment for balance conditions, developing a new test for the balance system. But I'm also really passionate in Alzheimer's research. So I'm hoping to, whilst during my PhD, which I'm doing alongside the clinical trial and the clinic work, I'm hoping to compare concussion and Alzheimer's disease and their various pathologies. Um, what was your other question? Sorry, was it around the latest research that we're doing? Uh,
Yeah, yeah.
yeah. So the most recent work that I took to ECSS in Rimini was our research that we collected in the clinic. So we have created the first UK university concussion clinic. And with that, we were able to not only provide a great clinical practice for our athletes to utilise, where we've got our trained physiotherapists that are able to diagnose concussions or and determine whether it's safe for our athletes to go back to play. But we can also apply that to our research setting and approach these athletes and see if they want to take part in the various studies that we've got going on in the School of Sport and Exercise Sciences. So one study that we've been carrying out for over 2 years now is where we've collected biomarkers at various stages of return to play. These biomarkers have been via saliva and via serum samples from blood. And I've presented what we found from our serum samples at ECSS. It's been quite exciting. Yeah, we've found some really exciting things with 4 biomarkers that we've looked at there.
Awesome. So two of those biomarkers are neurofilament light chain and glial fibrillary acidic protein.
Yeah.
So for our viewers, we'll call them NfL and GFAP from now on. I'm going to ask you about those proteins and why they're significant and what they tell us about brain injury. But is there anything you can tell us about those proteins in general? Like, what are they? If we weren't looking at concussion, what are they normally? How would you describe them? And can you just explain that, just so that our listeners are not all as smart as you? Even I'm not.
The reason why these 2 proteins in particular are quite exciting is because neurofilament light chain is released as a result of axonal damage.
Okay.
So when you've got a neuron, you've got almost the main body of the neuron at the top, and then there's it's like, trying to think of a good way to describe it, like a wire that leads down to the other end of the neuron. That's called the axon. And so when that's damaged, could potentially be as a result from a concussion or a head impact, then neurofilament light chain is often seen. And so that's released as a result of that damage. Now, GFAP, or glial fibrillary acidic protein, it's quite a mouthful, isn't it, to
Yeah.
say that one? I tend to just roll with GFAP, otherwise I'll get a bit tongue-tied.
No,
Um,
GFAP is okay.
we often see that. Uh, so we've got another really abundant cell in the brain. In fact, the most abundant cell in the brain is astrocytes, and so
Yeah.
they were originally called glial cells, and that's because originally years ago they thought it was just the glue that held the brain together. But it turns out these cells are actually really, really important for brain's function, and they provide energy to neurons. So neurons can't function on their own without astrocytes. They carry out the aerobic respiration to supply the neurons with energy. Otherwise, they'd run out of steam pretty quickly. So
Right.
now when astrocytes become activated and unhappy, they release GFAP. And so that's also why we were interested in these two proteins, because they're two cells that work together. And so these proteins, it's quite important to look at them both at the same time.
Okay. So in the study, your recent study, I think it was in 2022 that it was released. So the results showed there that NfL levels, so neurofilament light chain levels, decreased throughout return to play, but GFAP had a more acute response. So what does that tell us about the timeline of brain damage and recovery after concussion, as much as you're able to kind of deduce at this point?
So in our most recent study, so this, I think you might have looked at my poster from ECSS and you've
Okay,
read
maybe I'm
about,
thinking of the wrong one.
yeah, so you've read about the previous research. So there have been two studies which have also carried out Samoa analysis, which is what we used to analyze our blood samples. And we were quite perplexed at first when we looked at our samples because we thought, oh, we found a different pattern here with our samples. And we think that it's come down to the timing of when we took these samples. And so we think the timing of the sample post concussion is going to be really important to determine almost the severity of the head impact. We need to do some more investigating into it, but that's the exciting thing for us. And so what we saw was that GFAP hadn't increased, whereas other studies have seen that GFAP significantly increased post-concussion. However, we saw that NfL had increased, and other studies haven't seen that. They saw the opposite. One study saw something similar to us previously.
Okay.
However, we think that GFAP has an earlier increase. So astrocytes might get damaged during the massive head impact from a concussion, which shakes the brain around. And then the astrocytes recover. And so we took our samples, perhaps say 7 days after on average.
Right.
So we've missed that instant peak, which other studies have picked up previously. But the peak that we did pick up instead was the increase in NfL. And the exciting thing about this is that astrocytes potentially have become activated as a result of the head impact, which has led to them not being able to support neurons efficiently. And so then we've picked up this increase in axonal damage later on. And what another study found previously, was that the more increased NfL was post-injury, the longer it took to return to play. And so it's almost like, can we capture whether a concussive event has occurred pitch side by identifying the level of GFAP and then follow up with them in a few days' time to see if this has led to axonal damage. And it's so it's almost like the two, but we have to wait to see whether it's been severe enough and whether they're ready to return to play. Yeah.
Okay. If you allow me, I'm going to try to summarize and give you a repeat back here. So there's an impact to the head, and what you're saying is that maybe what you're thinking here is that the sudden impact to the head causes damage to the what used to be known as the glial cells,
Yeah.
but they're now known as astrocytes. The astrocytes are what normally support the cells, and so since they're busy dealing with the impact, doing whatever it is they have to do, you're saying that they're not able to do as much of their normal function. Sort of like their capacity has been taken up dealing with the impact, such
Yeah.
that down the line from that 7-11 days down the line, you're seeing the reverberations of that. You're seeing impacts down the line of the axons being able to actually transmit information, or markers that would suggest that they're having difficulty transmitting the
Exactly.
information. So there's the impact originally. So now what you're thinking is, if we can get these biomarker tests done earlier, we might be able to figure out if that is true, if there is actually a spike and then a
Yeah.
tail sort of.
Exactly. And that's why it's really exciting. But also it coincides with, so sometimes you get an athlete who's hit their head but
Right.
thinks, "Oh, I'm completely fine." And so when you do the SCAT test, pitch side, they're showing no symptoms. And then what we've seen often is actually in a couple of days' time, the athlete's thinking, oh, I've got quite a bad headache now. Oh, I'm quite sensitive to light now. Oh, I feel really, really foggy. I don't think I'm going to be able to do this assignment because I'm just staring at my screen and it's driving me crazy.
Yeah.
So it makes sense that neurons are actually okay on their own for a little bit. And then when the astrocytes are still recovering, um, but they're still not supporting the neurons sufficiently. And so we get this delay in symptoms, which is what makes second impact syndrome so dangerous, because they think, actually, I'm okay to help out with the second team a couple of days later. But then they're actually still concussed. It's just their symptoms haven't started to show up yet.
Right.
But it also means that all hope isn't lost. So say if we were to miss that GFAP peak, if they were to rock up in a clinic and we could see that increase in NfL, we could potentially still pick up a concussion, because whatever solution science comes up with, it's got to be practical. And
Mhm.
we know that, say, grassroots sports won't be able to do a really sophisticated, instantaneous pitch side test on biomarkers. A lot of the time, people have to go for a blood sample and it has to be processed, and it takes a couple of days to come back. Whereas this, allowing it to be detected in serum in particular, because serum can be transported. Whereas if we were to take it from plasma, plasma has to be stored on ice straight away and processed almost instantly. And so we're trying to come up with a practical solution. And this is another reason why the delayed peak in NfL might offer a more reasonable solution for athlete care going forward.
Right, definitely could be something that you look at throughout the return to play process. I think when I think about concussion protocols, and I've been talking to a lot of people over the last 5 years, sports leaders that want to do what they can to protect their athletes, and typically these concussion protocols will have a 24, maybe even a 48 hour. If you got pulled off the field or the ice or whatever, you can't return because we need to do obviously an initial test, but we need to be able to do one again. And typically that's just like a standardized time frame. But it, I'm ashamed to admit that I always thought of that as maybe it's just adrenaline, right? I mean, is there like an epinephrine or a norepinephrine, like adrenaline type of response that could play into this as well, where athletes are feeling the adrenaline from the hit, and that's why they don't notice the symptoms as much for the first couple of days.
You
Um,
could be onto
is both
something there,
could both be true?
definitely. Well, yeah, that
Could be
could
related, I
definitely be
guess.
part of the. Because I mean, I suppose if you were to have a concussive event at home, you're not full of adrenaline. If you were to just walk into a door by accident, you'd probably go, "Oh, that really hurt," and it would stop you for a moment. Whereas, like you say, on the pitch, you don't actually notice how hard you've hit your head as well, or on
Right.
the rink, for example.
You might not even notice how hard you twisted your ankle, right? Like, I mean, that it happens all the
Yes,
time.
exactly. Yeah.
Interesting. Okay, so normally in a concussion protocol, just building off what I said before, you're doing a scat maybe on the a shortened version of the scat on the pitch side. Let's say if there was a soccer match, um, and then you're just continuing to basically do that assessment. Maybe you're doing some symptom evaluations. How close are we to being able to supplement that or replace that, or at least supplement that with objective biomarkers like NfL and GFAP for the real world return to play decisions? Is it just a cost issue, or is there just more work that needs to be done on validating it, or both?
I would, me personally, I'm excited, and I always feel like we're almost there. But then you don't want to rush anything and go into something too quickly. Like, for instance, this year we had a particular focus on female athletes because we wanted to double check whether any of the biomarkers were impacted by the menstrual cycle and the fluctuations in hormones. We know that progesterone can have a huge protective effect on the brain, and we want to understand whether that can impact the various symptoms that we're seeing, and the biomarker levels as well. We are working with a company called Marker Health, which is also on our campus. They've developed quite a robust saliva test. And so things are happening all the time,
Right.
everywhere. I do feel like we're nearly there. Um, but in our clinic in particular, we just want to make sure that we're going to represent all athletes, and we want to try and cover all bases and be as robust as possible. And so I can't say for certain whether we're there and ready to go, because also we've then got to think about how can we make this work for grassroots teams, because a lot of people a few days after a concussion will show up at A&E, and quite frankly, A&E doesn't know how to deal with concussion. So we've got to make sure that how are we going to introduce this, and how is it going to look after athletes in the UK? So, yeah, we need to correlate it with all sorts of things. Like, for instance, the SCAT6, one of the next steps for me is, because balance in the vestibular system is also an interest. I know the SCAT6 can be very subjective.
Mhm.
It's important that we understand what athletes are experiencing, but we can measure balance objectively. And so I'd like to implement into our clinic some more objective tests of the balance system. And we've just got lots more detail to look into, just because if you want to do something, we want to do it thoroughly. So
Yeah,
it's
no,
not
makes
much
sense.
of an ambiguous answer, I'm sorry. But
I don't
yeah.
know, that gives me an idea of how close we are, right? I think we're pretty
Mm.
close,
Mhm.
in summary. But so you mentioned just throughout that, you mentioned menstrual cycle phase, and its impact on symptom severity. Do you want to just tell us a little bit more about what you found there, and how that might, how those might impact concussion diagnosis, and kind of what the next step is for your research in that field?
Well, the next steps that we're taking with this is, so we tried in the first year of the clinic to monitor athletes' menstrual cycles, and we came we came to face reality of that. One, it's really difficult to get somebody who's not feeling very well, to track their menstrual cycle, for one.
Right.
And two, we're dealing with university athletes as well. So they're on various different kinds of hormonal contraception.
Mhm.
And the third being, the menstrual cycle is actually very different from person to person. So you've got some athletes who might have endometriosis, some
Mhm.
athletes who have polycystic ovary conditions. There's and and to be honest, not enough research has been done into female hormones, and not enough research considers female hormones previously. And so what I'm quite excited to do is, because we always aliquot lots of different Eppendorfs with the same sample, so that we can go back to them. We've got our biomarker data now. What I'd be really keen to do is go in and do hormone ELISAs on those blood samples, and understand actually what level of progesterone, what level of estrogen did they have, and is there any correlation, or is there any pattern that we're seeing in our biomarkers. And because even if they're all at the same phase, or they're all on the same hormone contraception, um, the hormone levels can still vary from person to person, and that's the next step that I'm really excited to do. Yeah.
Awesome. So the last question here about research, I guess, would just be, you've recently partnered with us, HeadCheck, a few months ago. How do you see our partnership helping you with current and future research initiatives?
I like, from our first chat, I was so excited when we were able to chat together, and you could tell me a bit more about the app, because we've always said in Sportex, and when we set up the clinic over in MoveWell, it has to be a clinic first. And so we needed a really robust tool that could collect this large data set, because it will be such a huge data set from the SCAT6, and that is the gold standard tool for concussion now. And we were dealing with paper forms, and it was making it so difficult to collect baseline data from our athletes. And so this is going to enhance the clinic so much with working with HeadCheck. And then you guys also have that tool where we can pull the data and compile it really quickly. And so it's going to not only enhance the clinical side for our athletes, it's going to enhance how well and how efficiently research can use the data and further research in this field. So yeah, I'm really excited to be working with HeadCheck on this.
Awesome. So yeah, tell us just for the clinic questions. There are a few more questions here. So can you tell us a bit more about the MoveWell Clinic, its mission, key focus areas, and why it was established?
Yes. I remember at the start of the recording, you said you're going to talk to Sophie about her clinic, but it's definitely not my clinic.
Oh,
There's
clinic
a
that you're
whole
working with.
there's a whole team of us, and I
Yeah,
certainly we couldn't do it on our own. Like
yeah.
there's like Professor Sam Lucas, who does so much heavy lifting for the clinic, and
Mhm.
also our lead physio Dan Walton, and we've had so much help from our student physios as well over in the clinic. It's created lots and lots of training opportunities for them. We've also had lots of other colleagues helping out, like master students. We've got Izzy, Cashmore, Asha Strom. She's recently started her PhD in concussion as well. Uh, and it's, yeah, so I just wanted to say that. But
No,
first,
that's great. That's great. My bad.
and the school's been so supportive with developing it. It's really exciting. We're going to have some professional dissertation students coming over as well, and my one of my other PhD supervisors, Raymond Reynolds, is going to get involved and try and enhance the balance side of things. Sorry, I went off on a tangent trying to talk about how amazing
No,
everybody
no,
is,
that's great.
but
That's great.
um, yeah. So, was it you wanted to hear
Well,
more about the clinic? Sorry.
yeah, overall, I mean, you've mentioned all the great people working with the clinic. What's the overall mission goal of the clinic, and what areas is it focusing on?
So, MoveWell as a facility has got so much potential. It's, I suppose, the overall goal is to, we don't want to run before we can walk because there's so many. The trouble is, on campus, there's so many opportunities, and being close to the QE, there's so many opportunities. That's the UK Centre for Trauma, so it's a huge hospital near the university,
Yeah.
just for context. And we work with a lot of consultants over there as well, and so we've got really great clinical oversight and lots of advice that we can take from individuals. So where I can see this going, I feel like the sky's the limit with it, because we want, for me as well, I think it's really important to, with the NHS struggling at the moment, I want to show how clinical and research can come together, and how that if clinics and medical practices are managed in a meticulous way that can work for research, we can not only enhance science in a more effective way, but also with it being managed in such a meticulous way, I think be we can provide a much better support for our patients. Because one thing in the NHS at the moment is a lot of people aren't communicated with, because quite it's so overrun at the moment, and so what we've done with this clinic is strip it back to the very bare bones and to go, right, what do we need to be able to make it work for science? And I think that's really helped. um, and what I'd like to do is, because it's set up in this meticulous way, we can then bring more research projects in and build it up from there. And so I've alluded to the fact that we're hopefully going to bring more balance equipment over there. Uh, we're developing on my other clinical trial that I'm working on, a new test for the balance system, which uses electrical stimulation. Um, that's quite exciting because we've synced it up with we record eye movements. And so if we can potentially use that to detect neuronal delay, I'll be very excited to apply that to our concussed cohort, especially since we've seen increases in neuronal damage biomarkers. Um, Sorry, I'm waffling on a little bit. It's just
No,
because I'm
no,
so excited about it.
yeah, that's great. So MoveWell is obviously innovative and obviously has a research focus. Is there anything else about MoveWell that makes its approach unique compared to comparable or traditional clinics in the UK?
I think it's got people who are on the forefront of research helping to
Right.
advise there as well. And we also provide opportunities for our students to train there. So we have now student physio placements, and we're going to have professional dissertations taking place there with our sports science students. Um, I know that when our students are away over the summer, Me and other colleagues have got plans to potentially open a dementia clinic there as well, which is where we're going to attempt to compare concussion and Alzheimer's disease. So I suppose that's another exciting part of it as well. It's putting everybody who's passionate about research and its medical applications and sports science, it's putting us all together in a little pot, and
Yeah.
we're able to just make things happen.
I'm curious, going off script a little bit here, but I'm curious about, you mentioned comparing Alzheimer's and concussion. What do you mean by compare? Like, are you looking for similarities? Are you thinking that they might be related? Both?
Well, so you've probably seen in the news a lot of athletes now who've had, like, footballers in particular and some rugby players.
Yep.
Like, there's one example of a rugby player an English rugby player who doesn't remember winning the Rugby World Cup. And there's a lot of correlations being made with repeat head impacts and development of dementia later on in life. And if you look at how both are diagnosed, they're both currently diagnosed with very subjective questionnaires. And if you look at the symptoms of concussion, so you get loss of balance. Don't always get it, but you get loss of balance, fogginess, difficulty with short-term memory, cognitive, being able to think of different words, different cognitive issues, headaches, nausea. And in the very early stages of dementia and Alzheimer's, we also see those same symptoms.
Hmm.
It's just there's a spiral of decline in dementia and Alzheimer's, whereas most athletes tend to recover post-concussion, albeit varied in its timeline. However, we're now seeing that the more concussions you have, the more at risk you are of developing dementia. And the way that dementia resonates in the brain, particularly in Alzheimer's, we now done some cell culture work recently where we grew astrocytes, and they were showing we haven't looked at GFAP in particular, but they were becoming activated very early on in the onset of dementia, and that makes sense with the loss or not being able to think of words correctly because they're not quite able to support neurons.
Mm.
Normally, the brain's very good at carrying out neuroplasticity and fixing itself. It's just there's this one little trigger that happens where, for some reason, the brain isn't able to fix itself anymore in dementia, and we don't know what that is. And if we can understand the difference between the two, that might open new avenues. If we can learn from concussion, let's say.
So traditionally, I'm gonna keep going on this just because I've been interested in this in a while. Maybe I'm out of the loop here. Last I looked into Alzheimer's, there was these things called neurofibrillary tangles and beta amyloid plaques. Um,
Yes.
what's the latest on that? I guess, and in the last few years, I know it's
We've
a really
got
broad question, but I just didn't hear you mention them. So
we've got neurofibrillary tangles and amyloid beta plaques. They're definitely hallmarks of Alzheimer's, and you'll see them on that. You'll see plaques, and often people will be diagnosed post an MRI scan, and you'll see the plaques present in the brain. And that's what differentiates Alzheimer's from other kinds of dementia is the amyloid beta plaques. However, when they removed the amyloid beta protein in mice models, the mice got brain hemorrhages. So that protein is still very important for brain health. And is there a step before that which leads to the brain? It's almost like
it's too late at
it's
that point.
almost like the bin men aren't coming to pick up these amyloid beta plaques,
Yeah.
So is there a step before that which we're missing? Because we know that before dementia onsets and you're able to notice these symptoms, it's actually likely onset about 30 years before.
Mhm.
Yeah, this is the hoping because we can now use iPSC-derived stem cells, which are human stem cells, with human Alzheimer's disease. In a lot of cases, we'll use familial Alzheimer's disease, which is people who have a genetic mutation, which means they'll likely develop Alzheimer's by the time they're 30 years old. And so thank you to them. Those people have wanted to help further research, and they've donated some stem cells, which we can then differentiate into various different cell cultures. And then we're able to look at the cell from healthy through to the very end of the disease. So, yeah. And I'd like to, obviously, I don't know how to create a concussed cell in a dish, but
Yeah.
it would be very interesting if we can understand the pathology behind Alzheimer's and see if it compares at all with the pathology of concussion.
Right. So probably understanding either concussion or Alzheimer's, dementia, any of those will help. They are related, and we will be able to help our understanding of all of them by helping understanding one of them, probably.
Yeah, the truth is the brain is such an uncharted area, such
Yeah.
a grey area of research, because it's so complicated and so magnificent in how it works, and we're used to it just being able to fix itself. And so when it starts not being able to do that, we've now got to use our own brain to figure out what's going on with other people's brains, and it's
Yeah. Awesome. Well, we're just past 30 minutes here. Just wanted to give you a chance to mention anything that you haven't mentioned yet, sort of like looking ahead. What are you excited about down the line for MoveWell? And is there anything else that I didn't ask you about that you really wanted to talk about before we wrap up?
Well, I'm really excited for the third year of the concussion clinic. I'm
Yeah,
excited now that we're not going to have to deal with reams and reams of scat six on paper.
save you some time.
Yeah, I'm quite excited to get using the app and having the baselines collected with all the athletes. And this year in particular, I'm keen to start implementing the objective tests of balance as well. So we've got the blood data. I'd like to understand whether this blood data can correlate with the balance tests. Yeah. So,
Awesome. Well, Sophie, it's been such a pleasure talking to you. Thank you so much for your time. Thanks to everyone listening. Once again, it's HeadCheck Health Talks. I'm Eric VanderHelm, and have a nice rest of your day.
thank you for having me.
Thank you.